Welcome to a short presentation on the topic of yeast transformation. This is for you who are doing a semester project in the Biotechnology laboratory. Perhaps you are going to do this experiment yourself, and you can see this as a brief introduction or a set of guidelines before going into the lab and trying out this protocol for yourself. Transformation is the process of altering the genetic content of an organism. So that means we can either add, delete, or replace a part of the DNA inside a cell. Today I'm going to walk you through the steps necessary to achieve this genetic alteration in our favorite microorganism, yeast. At the end, I'm going to give you a few examples from previous semester projects, and I'm going to link you a few research articles that you are welcome to read to start your literature search. First, I'm going to give a short introduction to the subjects. In biotechnology, we are interested in utilizing living systems and organisms to produce products that could be valuable to us. So one example is natural compounds. Many different natural compounds exist. They are relevant to medicine, pharmacy, and food additives. Here we see an example of carbonic acid. It's a red dye or a pigment that's synthesized in nature like this, and it's a very stable molecule that people have used for centuries in the textile industry and for coloring food. So carbonic acid is a naturally occurring pigment in these beetles, and they are living on these cacti in South America. Actually, 25% of the dry matter content of these cactus beetles is carbonic acid. So they are producing loads of this compound that is easily extracted and used in downstream processes. It is probably no surprise that customers are not happy being told that they have been eating insects all their life. So perhaps we should figure out an alternative production method of such a natural compound as carbonic acid. Perhaps we can utilize biotechnology in this instance. And sure enough, there is a biotechnological solution to this problem. Perhaps we can pick out the genes necessary for the biosynthesis of carbonic acid, and we can take these genes, we can copy them from their original host, and we can place those genes inside of a microorganism, which is then capable of turning on those genes, building the machinery necessary for the production of carbonic acid, and we can then scale up a biosustainable production anywhere in the world for carbonic acid. And this time you do not have to kill any animals, and it's no longer a requirement to be in the desert in South America. Furthermore, genetic engineering can contribute to a greener and cheaper production of this desired compound. And that is why we need to use transformation. So in transformation there is two parts. First, we need some sort of source of foreign DNA. And in this example, it's the beetles. And we are copying this small section of their DNA. This is also called cloning. And then we also need some fresh yeast cells. And then we need to make a critical step here where the cells are becoming competent. And why is that important? Well, you can't simply mix free DNA and cells and then hope for transformation to spontaneously occur. It's not going to happen. We need to treat the cells first, making them competent, making them ready to take up the DNA. And then we add the DNA in the transformation reaction. The transformation itself is what I've drawn a box around here. It is the introduction and take-up of foreign DNA, and that DNA can actually recombine itself with the genome of our host, and it is then fixed within the genome of the yeast cell, and we have now created a new version of this yeast, one that has a different genetic state than its starting point. It has been altered in that sense, and we call this cell a transformant, or sometimes also a mutant. So this mutant has received some extra genes, and those genes are also inherited by offspring from this cell. So this will be the first and single transformant that we create, and then from that we are capable of scaling up to an infinite amount of cells with the same genetic modification. Why do we choose yeast as the recipient cell of our genes? Well, yeast is an extremely popular expression organism in biotechnology. It's easily culturable, it's cheap, it's easy to transform, they are sustainable. It's a representative of eukaryotes, so like humans and plants and fish and insects, it's a eukaryote, but it is unicellular. So it's pretty easy to work with. It's not a multicellular organism like a human is, but it does contain all of the same essential molecular building blocks. So yeast has been used as a model organism for many studies. For instance, sexual genetics or DNA repair or aging and so on. And besides that, it's just an industrial workhorse. You might know it's used in beer and bread production, and you can buy these cubes of yeast in your supermarket for no money. I will now run through the protocol for transformation of Saccharomyces cerevisiae, which is the common baker's yeast. The method we are using was described by Daniel Geitz and Robert Schitzel in 2007, and it has been adapted by many labs and many research studies. I will provide a link to this publication at the end of the slideshow. For transformation of yeast, you can split the workflow into 4 categories. First, you need to grow some cells that you want to transform. Then we make the cells competent by treating them with a few chemicals. Then we introduce the DNA, so this is actually the transformation. And lastly, we recover the cells. And why do we need a recovery step? Well, treating yeast cells with all these chemicals is kind of toxic to the cells, so good recovery is necessary. I will now go through the transformation process of our favorite yeasts, Saccharomyces cerevisiae. The protocol we're adhering to here is developed by Geitz and Schitzel, and I will post a link to this publication at the end of this slideshow. In general, there are 4 major steps to this process. The first step is growing some fresh cells. We need to harvest them at the optimal time point in their growth. Then we perform the treatment with chemicals where the cells are made competent and they are ready to receive DNA. In the third step, we actually mix our competent cells with DNA and perform the actual transformation. And the final step is recovery. And why is that important? It's important because this treatment is actually very stressful for the cells, and we are adding a few chemicals that can be considered toxic. So we need to ensure good recovery of the cells in order for them to survive this treatment. The first step is, of course, producing some cells. And you will start out with a culture like this, with some fresh cultivation broth. and you will add some cells. After 4 hours, you should get a milky or an opaque culture like this, and then we need to harvest the cells. This has to happen at an optimal time point, and you can figure out when to harvest your cells by measuring the optical density of your culture. It's important to harvest the cells at the correct time point. Just to visualize it, I've added a small growth plot here. This is the number of cells and this is time. And you can see that we begin with a few cells and then they will start dividing. We have a high growth rate here in the exponential phase. And then at some point the cells will run out of either substrate or space, and the concentration of cells will remain constant. At some point we will even get cell death if they have been in the stationary phase for too long. We need to harvest the cells at a point in time where they are most likely to survive. And this is where they have a high growth rate. This is where they are used to taking up substrate and dividing themselves at a very high rate. So if we harvest the cells too early, you will get too few cells. And if you harvest them too late, you will be going into the stationary phase where the cells are more dormant and they are less likely to be transformed here. Here I have drawn a small visualization of a yeast cell. It comprises all the basic elements you would expect to find in a eukaryotic cell. The cell wall, shown in green, a cell membrane, it's the black thing. We have some cytoplasm in brown and a nucleus with the intrinsic DNA of the cell. The first step to making competent cells is removing the cell wall. The cell wall is very complex, it's very thick, and we treat them with a mix of enzymes that is capable of removing some of these complex structures. It is now only packed inside the membrane, the bilipid layer membrane. So they are very fragile, as you can imagine. So rationally, the first step we do is submerging the cells in a chemical known as polyethylene glycol, and these are long polymers that are capable of acting as a kind of synthetic cell wall for our fragile cells. It also immobilizes the cell, making them less susceptible to osmotic pressure. The second thing we add are lithium acetate ions, and lithium is very toxic to the cell. It actually interacts with the cell membrane and creates a porous structure in the cell membrane, making transfer between the cytoplasm and the exogenous environment possible. You can now think of the cell as a cheese full of holes. The last ingredient we add is single-stranded DNA from salmon testes. And whoever came up with this idea to add this particular chemical, I have no clue. But this is how it works. You see, DNA wants to make interactions with cell membranes. That's just how it is. But we are also adding DNA from our beetle. So we do not want our DNA to interact with the membrane. We want our DNA to be able to enter the cell. So to eliminate the possibility of our exogenous DNA adhering to the membrane of the cell, we add a lot of salmon sperm that contains many DNA molecules, and they will make interactions with the cell membrane here, blocking off the possibility of our genes adhering to the outside of the cell. And now we are ready to add our exogenous DNA. And in the example I mentioned before, these would be the copied pieces of DNA from our beetle. So This time our DNA is capable of entering through the opening in the cell membrane and entering the cytoplasm of the cell. Finally, we need to recover the cells, and we do that by washing away all these chemicals, especially the lithium, which is toxic to the cell. It is not healthy for it to be in contact with lithium for so long, so we need to rinse that away. And we now give the cells time to recover and repair their membrane and build a new cell wall and so on. We'll end up with a cell with a repaired membrane and a repaired cell wall, and our exogenous DNA is now capable of recombining with the genome of our microorganism. As a final example here, I'm just going to show you a few plates from previous student projects. This is actually the yeast in our laboratory as it looks in its natural state, and we then introduce a piece of DNA encoding for the biosynthesis of this orange pigment. And it is very easy to see that the yeast has changed phenotype from pale white to orange. Another example can be seen on the right, where the pigment is a bit more yellow or brown. So you can do this yourself. If you could choose which biosynthesis pathway, which product, which gene would you like to select in nature and put into your favorite microorganism? If you want to know more about yeast transformation, I highly suggest that you start by reading the following papers listed here.